Hello students, you are welcome to the session 9 of this course Energy 207. In this session, we are going to discuss bacterial pathogenesis. When you talk of pathogenesis, what does it mean to you? Look at the two basic words there. Toxin means toxin or poison. Genesis means the beginning or production. And this stands for ability to produce toxin. This is an underlying mechanism by which any bacteria pathogens produce disease. At a chemical level, there are two main types of bacterial toxin. They are called lipopolysaccharide, which are associated with the cell wall of gram-negative bacteria, and protein, which are released from bacterial cells, and the mineral tissue site removed from the site of bacterial growth. So these are referred to as exotoxin. As we go along in this course, understand those words very well. When you have started this session, you should be able to differentiate between types of bacterial toxin, to explain bacterial protein toxin and their properties, to describe mode of action of bacterial toxin, and last factor that control and regulate synthesis of bacterial toxin. Let's look at bacterial toxin now. Type of bacterial toxin are endotoxin and exotoxin. When you say endotoxin, they are cell-associated substances that are structural components of bacteria. Most endotoxin are located in the cell envelope. That is, endotoxin are located within the cell envelope of the bacteria. They are part of the bacteria itself. Like I have said, they refer to lipopolysaccharide or lipooligosaccharide located in the outer membrane of gram-negative bacteria. Although components of cells soluble in endotoxin may be released from growing bacteria or from cells that are alive as a result of effective host defense mechanism by the activity of certain antibiotics. Endotoxin acts in the vicinity of bacterial growth or presence. That is endotoxin for you. Endotoxin produces fever. It produces leukopenia. It produces shock. And they are product in the activation of complement system under immunity. And let me say that from this shock, death may occur due to shock or DIC. DIC is known as disseminated intravascular coagulopathy. When you talk of exotoxin, they are secreted by bacteria and act at a site removed from bacterial growth. However, in some cases, exotoxin are only released by lysis of bacterial cell. They are usually protein, minimally polypeptide that act through direct action with host cell and stimulate a variety of host responses. Most of these toxins act at tissue sites, removed from the original point of bacterial infection or growth. However, the bacterial toxin act at the site of pathogen colonization and may play a role in infection. Example of this is when one is infected with tetanus. This Clostridium tetani may enter into the body at the foot, that part of the body where the wound is located, but will not act on that site. The effect will go to the neuron, and because it has neurotoxic effect, that is exotoxin, because it has made up the fact that the toxin produced does not act locally, but we have to travel to a far distance in the body before it can produce its toxic effect. Now, we look at properties of protein toxin. How do they act in the body? The first thing they do is invasiveness. They produce one enzyme they call infesin because they act locally to promote bacterial infection. When they do that, when they produce infesin as an enzyme, they will now have what it takes to enter into the cell of the host. That is what they call invasiveness. Second characteristic they have is they are specific in nature, because those that we attack a particular type of cell cannot attack other cells. They are meant for that cell. Like I said in case of Clostridium tetanus now, by the time it enters the body, the area it's going is the neurons. And until you get to that neuron, it could not do anything. That is what they call specificity. That is, it is specific for a particular organ, and it not go to another organ than that organ is meant to act on. The third one is what we call antigenicity. Antigenicity. Because the toxin produced by this organism, they are antigen in nature. And when they enter into the cell of the host, they must be able to stimulate antibody reaction. So this is a specific property of protein toxin, that is, they are antigenic in nature. They are antigen, and that antigen must play a role in the host by producing antibody that will work against the effect of that antigen. The fourth property is what we call instability. This protein toxin, they are unstable. They lose their toxic property but retain their antigenic one. That is, when you see a toxin, they have two properties. They have protein properties and they have toxic property. But when they lose their toxic property, they retain their antigenic property, which means that they can be more virulent when they pick up another toxin. And this is what was used to produce tetanus toxoid. Because when the man called Ehrlich discovered that tetanus organism has both antigenic and toxic part, but can easily lose its antigenic, can lose its toxic part, retain only antigenic part. That toxin was now used to make a vaccine, which is now used as a vaccination against tetanus infection. Because of the fact that it retains its antigenic activity, it is immunogenic in nature. It has immunogenic capacity. So that is why this is used to treat tetanus in clinical setting. So instability is a relative term that makes the toxin of this protein unstable, because they lose it but they retain their antigenic property. When you look at the mode of action of protein toxin, one, they have attachment and entry of toxin. They can attach to the cell of the host, and the ability of entry into that host. That is the first property they possess. Second one is pore-forming toxin. They have a pore-forming toxin in the way that, like you have in some of these bacilli, they are pore-forming. That is why they cannot be destroyed easily. So when you have protein toxin and a pore-forming toxin in nature, it means that toxin they are producing cannot be easily destroyed. So that is why that they can be easily destroyed even with utmost form of sterilization. So they have attachment property and they have pore-forming toxin. They can attach to the cell of the host, penetrate into that cell, producing their toxin inside the host. and they have performing toxin on the cell of the host. This toxin when they produce will start weakening the cell wall of the host, then that will allow the toxin to enter into the cells. This diagram shows you the attachment and entry of toxin in protein toxin. You have what they call direct entry. That is the B subunit of the native A and B toxin bind to a specific receptor on the cell, which induces formation of a pore in the membrane through which A subunit is transferred to the cell cytoplasm. So this is the B unit. By the time this B unit binds to a specific receptor here, it induces formation of a pore in this unit, and then we now allow the A unit here to form a pore that will enter into these cells, thereby releasing the toxin. This is the pore forming toxin that is seen. This is pore forming toxin. These are receptors. These are the cells that are being produced. So the toxin produced, we find a way of entering into the cell and clustering together the cells, making them inactive. That is what they call pore forming toxin. Students, you have seen how bacteria produce toxin. You have seen types of toxin produced by bacteria, and you have seen how this toxin actually work to produce disease in human being. By the time we come back to the next study session, we will go on with another interesting topic in our darling program, NSC 207, Medical Microbiology and Pathology. Until then, I see you in the next class.